All right. So there are basically three components to hemostasis, starting with vascular spasm. And vascular spasm is the vasoconstriction of vessels after they've been injured. After there's injury to the endothelium, there's exposure of the endothelium. Now we're going to trigger vasoconstriction in order to decrease the amount of blood loss at that site. The next step in that is going to be the platelet plug, which is where we have platelets adhering at first. We'll talk about platelet adhesion, and then platelet aggregation, which is the sequestration of additional platelets to create a platelet plug. And then the third and final step is a clot formation. So the aggregation of various clotting factors that we'll talk about, there's the clotting cascade, several clotting factors that are activated in series that brings about the ultimate fibrin mesh that makes a blood clot. Okay, and that is actually going to bring about hemostasis. It's going to block the site of injury, prevent any additional blood loss, and then begin to heal that exposed or injured endothelium. So let's go over some of the details of this. A lot of factors involved here and a lot of various steps. So let's talk about vascular spasm first. So after we have injury to a blood vessel, let's say you have a paper cut or a larger injury, you damage your endothelium, which is what causes the bleeding, right? That damage triggers vasoconstriction, which is to minimize the blood loss. Again, the body is very smart, very intelligent in this way. It wants to prevent not only healing the injury, but actually preventing excess blood loss. So automatically you trigger vasoconstriction, which then triggers the intrinsic vascular response, along with extrinsic responses that engage the sympathetic nervous system. So local factors and external factors are both working to minimize blood loss through vasoconstriction. And the second step to that is that the endothelial layer becomes sticky. We'll talk about von Willebrand factor, which is what sticks to the collagen, right? When we injure a blood vessel, the underlying collagen is exposed. That's in the underlying basement membrane. That triggers von Willebrand factor, binds to that collagen, and then now there's a sticky surface to which platelets will begin to adhere. So that sticky endothelial layer is also a part of the initial vascular spasm. Now we're going to work to form a platelet plug. And, platelets are thrombocytes. They are basically cellular fragments, so they are not cells. And if you think about the cell lineage that they're derived from, they come from a megakaryocyte, a very large blood cell that basically explodes, right? It explodes into many pieces, and those pieces are called thrombocytes or platelets. And they're very important in terms of aggregating at the site of an injury to a vessel. So they decrease the blood loss, one, but then they promote the various factors that help to form a blood clot that's going to be a more permanent plug to heal and then will permanently stop that blood loss. Platelets are colorless. They again are fragments, so they don't have nucleus. They do have organelles and granules, right? They can secrete certain secretory products that are going to further aggregate more platelets in a positive feedback loop. So they don't have a nucleus, they're not a cell, but they're cellular fragments that do have the ability to secrete products, and then they do have internal organelles as well. Now, in terms of the amount of platelets, we have anywhere from 100,000 to about 500,000 platelets in a milliliter cube of blood. And this is basically a very small amount of blood, about a teaspoon of blood. And so many, many, many platelets are found circulating in our blood, but they only make up about 1%. When you look at the overall distribution of the blood, which was covered earlier in this section, about 45% of that is cells, about 55% of that is fluid or plasma, and then about 1% of that is going to be leukocytes, which are our white blood cells, and platelets, which are our cell fragments. But they are very crucial, very, very crucial in blood clotting. So let's look at what this platelet adhesion cascade looks like. First off, we have blood vessel damage. And again, the details of this is that we injure the endothelium. So we expose the basement membrane, which is underneath the endothelium. And that exposure of the subendothelium allows von Willebrand factor to bind to the exposed collagen fibers of that blood vessel. Okay. Now, when von Willebrand factor binds, it creates a sticky surface that now attracts platelets to bind to that von Willebrand factor. This encourages platelet adhesion, which is the first sticking of the initial layer of platelets, which further creates another sticky layer. It's going to trigger platelet aggregation. And then it also secretes different factors that further aggregate platelets in a positive feedback loop. So adhesion, a very sticky surface, and then secretion of products and factors that further aggregate more platelets. And so von Willebrand factor is another important component in blood clotting. We'll talk about some of the clinical connections at the end, but von Willebrand factor is one of those. If we have an absence of von Willebrand factor, that can lead to various bleeding disorders, and that can lead to, just easy bruising, easy blood injuries, bleeding into the joints, various types of bleeding disorders. Let's get into platelet aggregation. So we talked about adhesion. Adhesion requires von Willebrand factor binding to collagen and then creating that initial sticky surface. Aggregation is what happens when we have platelets beginning to stick to that von Willebrand factor. So now platelets are going to stick. They're going to secrete several secretory products that further aggregate more platelets in a positive feedback loop that begins to create our blood clot. So first off is ADP, adenosine diphosphate. And this increases the stickiness of that first layer of platelets, and then it calls more platelets to stick to that area. It also secretes serotonin, which causes vasoconstriction. Epinephrine also causes vasoconstriction, and then other chemical factors that are going to facilitate more coagulation. And coagulation is just clotting of the blood to create a clot, right? To create a plug. We also produce thromboxane A2, and that can also further excite or encourage platelet aggregation. Now, what prevents the clot or platelet plug from spreading, right? It's not a clot at this point yet. I'm using the word clot very loosely, but it's a plug at this point. It's a temporary clot, which is just stopping the blood that will eventually be turned into a clot. So the platelet plug here is prevented from spreading to healthy areas of the blood vessel by two factors. Prostacyclin, which is PGI2, and that is going to be secreted by healthy endothelial cells, and nitric oxide also secreted by healthy cells. So this prevents platelet plug, which is adhesion, and platelet aggregation in the parts of the endothelial lining that are healthy, that are not injured, that do not have collagen exposed. All right, so let's look at that analogy here. So here we have a site of injury, right? Here's our blood vessel, here's the lumen, here's the underlying endothelium, and then here is that site of injury where we have the initial platelet adhesion. So the adhesion part of this is von Willebrand factor binding to the exposed collagen under here, and that begins to create the sticky surface for the next step in the process, which is aggregation. Now we have platelet aggregation, and that secretes two factors. It secretes thromboxane A2 as well as ADP, and those two factors encourage more platelets to bind in a positive feedback loop. We also have things like serotonin and epinephrine being secreted that cause vasoconstriction of the vessel. All right, so this is what is happening at the site of the injury. At other areas of the vessel, right? This can be either adjacent areas that are not damaged, such as these areas over here, or this can be the site of injury after it's been healed. We're going to have secretion of prostacyclin and nitric oxide that are going to prevent or block platelet aggregation. So at healthy parts of the vessel where there's no injury, there's no exposure of collagen, we're going to have nitric oxide, PGI2 that are going to prevent this platelet aggregation.